Today, we will look at the pathway of Alzheimer's and several ways in which scientists have tried to understand the causes of Alzheimer's in order to slow down its progression. Alzheimer's is the sixth leading cause of death in America and third for older people, with the average cost of treatment being $57,000 a year. Alzheimer's disease is also popularly portrayed in media, including movies like The Notebook, Still Alice, and shows like Grey's Anatomy. Usually what comes to mind about AD is dementia, as well as the differences in size of the brain compared to the healthy brain. Not too many people know, though, that the changes in the brain of AD patients occur due to the abnormal clumps of amyloid plaque in tau tangles. Brain damage starts at the hippocampus and spreads outward, which leads to cognitive decline. Various diagnoses lead to research focused on finding treatments that help reduce amyloid production, facilitate its clearance, and prevent amyloid beta aggregation. Scientists have also tried to research the various brain structures that could help healthcare providers and patients better understand Alzheimer's disease, which brings us to this.
Advancing our understanding and treatment of Alzheimer's disease begins with understanding the different brain structures that are affected, including the blood cerebrospinal fluid barrier, CSF for short, and the blood-brain barrier, BBB, previously researched by two researchers, Rosemary Vandenbroek and Matthew Campbell, respectively.
The blood-brain barrier, BBB, helps maintain the brain's homeostasis, brain stable state, but also complicates clearance of detritus, including the amyloid beta peptides. The blood CSF barrier is where molecules pass through and gain direct access to the CSF, cerebrospinal fluid, as well as the brain interstitial fluid.
In the Vandenbroek study, the researchers determined what effects AB oligomers have on a barrier between blood and brain that is under radar. That barrier is known as the blood cerebrospinal fluid barrier. The researchers observed the changes that the AB oligomers underwent in respect to its morphology and loss of BCSFB integrity, which could possibly induce the progression of Alzheimer's.
Campbell and his team used previous research revealing that the presence of amyloid beta in the brain causes suppression of tight junction proteins like occludin and claudin-5 to investigate the extent to which permeability of the BBB is affected when these proteins are suppressed. They used siRNAs to turn off expression of these two proteins in the paracellular gap, and they assessed the movement of small molecules of amyloid beta species across human Alzheimer's brains and TG2576 mouse brains.
Research led by Vandenbroek focused on harmful effects of amyloid beta on the choroid plexus epithelium, shown in Figure 1. The choroid plexus epithelium is a membrane that lines brain ventricles and separates blood vessels from the cerebrospinal fluid. After intracerebroventricular administration of the amyloid beta oligomers to mice, gene expression analysis of the hippocampus revealed that several inflammatory genes were significantly upregulated 6 hours after the injection of the oligomers, as shown in Figure 2A through C. The scientists found that the oligomers induced inflammation in the brain, choroid plexus, and cerebrospinal fluid. Amyloid beta oligomers were also found to be linked to increased expression and activity of matrix metalloproteinases, or MMPs. MMP activity was shown to increase in the cerebrospinal fluid 6 hours after oligomer injection, as shown in Figure 3B. MMPs are enzymes that chew up proteins, including those in tight junctions. Co-injection of amyloid beta oligomers with the broad-spectrum MMP inhibitor showed that the inhibition of MMP activity prevented the disruption of the blood cerebrospinal fluid barriers. Campbell's research team observed the size-selective movement of the amyloid beta peptides across the cell monolayers within the mice's brain endothelial cell. Using a t-test and densitometry, it was found that when the claudin-5 and occludin levels were decreased, as opposed to its natural 100% levels found in the mice with NT, which is non-targeting siRNA, based on Figure 1b, the amyloid beta 1 through 40 monomers were able to pass through the weakened blood-brain barrier. The gaps were only large enough to let the molecules of less than 8 kilodaltons to pass. Based on Figure 3, administering the two tight junctions siRNAs enhances the paracellular movement of the amyloid beta 1 through 40 peptides from brain to blood in the TG2576 transgenic mice. In the mice that received the two tight junctions siRNAs, displayed a significantly increased percentage of alternating behavior, with a mean of 68.51%, compared to the non-targeting siRNA-treated mice, with their mean being 62.54%. The percent alternation measures the spatial memory, since the mice have to remember that one of the arms contains a reward. The mice must recall their previous unsuccessful path to attempt an alternative method of reaching a reward.
Figure 6A of Campbell's team's paper visually depicts suppressed tight junction proteins in the brains of patients with varying disease. Compared to patients with brain injuries and ALS, Alzheimer's patients have more amyloid beta proteins, which are stained green, and less claudin-5 and occludin proteins. Figure 6B shows the number of claudin-5 and occludin protein vessels per millimeter of brain tissue in patients with a variety of neurological diseases compared to Alzheimer's patients, who had significantly fewer of these tight junction protein vessels per millimeter.
In the Vandenberg study, the main objective was to determine the effects that AB oligomers have on the blood-cerebrospinal fluid barrier in the brain. After a single intracerebroventricular injection of AB142 oligomers into a rat, the affected matrix metalloproteinase 3 gene altered the protein expression level of tight junctions, causing an increased leakage of a plasma substitute, dextran, from the blood into the CSF. When this injection was used on a rat that lacked the MMP3 gene, the leakage of dextran was significantly reduced. The use of a broad-spectrum MMP inhibitor blocked the effects of the AB142 injection, proving that the MMP3 gene is related to the inflammation of the choroid plexus epithelial cells found in the BCSFB. Thus, the study was successful due to the findings that AB oligomers are linked to the loss of tight junction functionality.
The amount of amyloid beta present in the brain can determine the expression of tight junction proteins. Campbell and his team found that more amyloid beta in the brain corresponds with less claudin 5 and occludin in tight junctions. This study is unique compared to other studies on the subject because it focuses not on transcellular movement, but specifically on paracellular movement between endothelial cells in the brain. Taking into account the inverse relationship between the presence of amyloid beta in the brain and expression of tight junction proteins, Campbell's team suggests that we can experimentally manipulate the composition of tight junction proteins like claudin 5 and occludin to create a controllable level of permeability, thus facilitating diffusion of specifically sized amyloid beta peptides from the brain to the blood. Campbell's group hints at the possibility of altering the BBB's permeability based on size of amyloid beta molecules. This could tempt individuals to try and alter the barrier so that the amyloid beta polypeptides can pass through the barrier, but this could allow toxins to enter the brain. Instead, perhaps future research can focus on ways to reduce the size of amyloid beta polypeptides so that they can meet the size of molecules that can pass through paracellularly, as Campbell's team found.
Connecting the two articles, we see that the blood-CSF barrier is more vulnerable than the blood-brain barrier, where the molecules pass through different boundaries to get direct access to the CSF in the brain intestinal fluid. Vandenberg thinks that the idea of Campbell's work is interesting, but is wary of the consequences of bad toxins entering the brain as well.
Results of the experiment support a past study which has indicated that the brain's inability to let buildups of amyloid beta leave the BBB could be a major contributing factor to onset of Alzheimer's disease, because Alzheimer's disease patients compared to healthy individuals had a lower rate of removal of amyloid beta 42 oligomers.
Though Alzheimer's disease is an irreversible disease, each of these papers looks at how alpha-beta proteins build up abnormally in different barriers in the brain and how we can potentially alter these barriers to control disease progression. With all this in mind, we see that controlling tight junction proteins and alpha-beta plaque is the key to slowing the progression of Alzheimer's disease.
Thanks for listening. We hope you enjoyed our video.